Hi, everyone. Nice to meet you. As Chris said, my name's Ken Burch. I'm currently a professor at Boston College, and my group is working closely with Grip Molecular on developing a new set of point-of-need sensors. So we call our system the GEMS, or the Graphene Electronic Multiplex Sensor. So I'll start today by telling you a little bit about sort of what's the problem we're trying to solve, namely that we want to develop new point-of-need sensors, and that there's always this kind of a multi-dimensional problem we have to solve of trying to get a sensor that can be specific but simple, can detect multiple analytes, but also have a low limit of detection, and yet, of course, balance that with cost. And I'll try to argue to you today that graphene is an excellent solution to that, because it is not only biocompatible, but it's very easy to attach a wide variety of biological probes. And it also has electronic properties that are very handy when doing sensing, as I'll explain. I'll then give you actually two examples where we've tried to detect different proteins that are important for biological problems for individuals. So namely, the first one is a protein that is indicative of the presence of disease. And the second one is where we've tried to detect antibiotic-resistant bacteria. So as I think everyone's well aware, when we cough, it's important to know what we're infected with. And so we're trying to develop point-of-need sensor that can tell you what is the cause of the infection, whether it be whether it be bacteria or it be virus. I'll then, at the end, tell you about another recent work we've done with the same platform, which I'm showing you here, which is the size of a penny, and can currently detect 4 different analytes, where we've been able to detect the presence of opioids in wastewater, showing that this works not only for personalized medicine, but also for pandemic level or sort of population level problems. Okay, so with that, let me jump into what the problem is. So sensors are quite ubiquitous in our lives, in all sorts of things that we might want to detect, and whether they're biological or temperature sensors or other kinds, they all sort of have the same problem that we're trying to solve, namely that we would like them to be simple to use. We'd like them to be specific so that they are detecting the specific element that we're interested in, so the specific protein, for example, and not sort of giving us false positives. We want low limits of detection, so we're as sensitive as possible. Ideally, we'd like sensors that can detect multiple things. So for example, it'd be nice to have your sensor that can tell you what the temperature is, but also what's the humidity in the room. Or in our case, whether it's COVID or a bacteria. And all of this, of course, must be balanced by cost. And this sort of balancing act, which is quite delicate, is one that, as an academic, I'm actually quite used to. But I hope to show you that graphene is a good solution for. So the particular version of this that we're interested in are what are called point-of-need sensors. So these are sensors where you're trying to get the detection out of the lab and to the point at which you need to know the information you're interested in. So in the case of individuals, right, you don't want an individual to have to go to the doctor's office just to find out what they're infected with, or even just for general laboratory measurements. It'd be nice if people could do it at home to take advantage of telehealth, for example. On the other end of the spectrum, we also would like to know what's going on in a population, and it doesn't necessarily have to be a whole city's worth. It might be nice just to know what, for example, on a campus is going on in a single dorm building. But nonetheless, we need sensors that make that possible. So the sort of cutting edge technology right now is something called PCR. And you could buy this multi-million-dollar machine called the BioFire, which can detect 21 different analytes. It's a pretty impressive instrument, and basically what it does is take DNA and essentially copies it many, many times, and then tries to detect it by the presence of specific probes to detect certain strands of DNA. And so that requires fluorescence and a very careful set of techniques and controls, as well as chemicals. And that's why the BioFire is such an expensive instrument and has to be run in a lab and by experts. On the other end of the spectrum, I think we're all pretty used to these rapid flow immunoassays. So these are basically strips where you put down